All right. It is 8, and let me turn off the waiting room. Welcome to the first session of Biochemistry 1, bright and early at 8 a.m. No, I did not pick 8 a.m. as a start point myself. Uh, I would not do that to you, but someone else did. So we're going to do it at 8 a.m. My name is Dr. Casey Andrews, and our plan for today is we're going to go over the syllabus. We're going to go over the Blackboard site, and then we're going to get into it because our classes are 2 hours. So unfortunately, I can't just spend the whole first day going over the syllabus. So for our meetings, generally what I do is that I will just blanket mute everyone just so we don't have background noise. If you have questions, feel free to put it in chat. If you are more comfortable speaking it, what you can do is either put in chat like, "Hey, I have a question. Can I unmute?" Or can I do one of those raise hands in Zoom, and I can also unmute you. And also about the chat, if you want to ask a question but you don't want to like have the rest of the class look at your question, feel free to send me private messages via Zoom as well. I will read those out loud if they're applicable to the class, but I won't attach a name to it. So only I will be able to see those names. All right, so what I'm going to do now is I'm going to share our syllabus, which should be available to you on Blackboard. And by the way, this is being recorded. So if you didn't quite catch something the first time around, I'm going to put all these up onto YouTube. All right, so Biochem 1. To contact me, the best way is via my email here. And I'll get back to you as soon as possible usually try to do that during working business hours. Just be aware if you email me like in the evenings or like early in the morning, I might not get back to you. If it's in the evening, maybe till the next day. If it's early in the morning, not until like working hours. Office hours are given here, but I will say that if these times don't work for you, just send me an email and I can meet pretty much whenever. I am teaching another class on Tuesdays and Thursdays. And that's from 11 to 2, so I won't be available then. But anytime in the morning, Tuesdays or Thursdays, or anytime after class, Monday, Wednesday, or anytime Friday, if you want to meet, we can set something up there, or just send me questions via email. Class hours, Monday, Wednesday, 8 to 10. So our textbook is Vote Pratt, and I just noticed that this is actually a holdover from when we used to do this. However, you might get the textbook for free. That's something we have to check because for some reason on Blackboard, you're not supposed to get the book for free, but last semester when I made it available to people, they were able to get it for free. And since that was the case, I didn't actually let the publisher know, hey, we have a free textbook open. So we're going to try the same thing again. If you have access to the textbook, and I'll show you how to do that on Blackboard, great. You don't have to do anything else. If they have revoked my free privileges, then we'll work from there and I'll tell you what to do. Homework is called Wileyplus, and I'll tell you how to access that when we go to the Blackboard site. Calculators, you're going to want a calculator. You don't need a fancy graphing calculator or anything. You just need a scientific calculator. And what a scientific calculator is just a calculator that can do like log or natural logs. So if you have a calculator and if you look at it and you have a button that's like X2 or you have a button that says log, that's perfectly fine. If your calculator is just plus, minus, times, and divide, that is not sophisticated enough. Scientific calculators can be bought for like $13, so they're not too expensive. Right, so what are we going to get into for Biochemistry 1? So here at Texas A&M San Antonio, we have biochemistry in 2 semesters. Other universities rather, have it in 1. So what's the difference between a 2-semester biochem and a 1-semester biochem? In the 2-semester, we really break up our topics. So the first semester is we are just looking at fundamentals. We're looking at biomolecules. We're looking at how these biomolecules come together to form polymers. We're looking at the basics of enzymes and just basics biochemistry of the cell. If you're inclined to do the second semester, which I would recommend if you are thinking about taking the MCAT or PCAT or exams like those, the second semester is when we're really going into glycolysis and all the cycles. So we won't be looking at those cycles this semester. Here, we're just going to introduce everything and really hammer home the basics. And here are like some of the topics. Like I said, we're going to look at the building blocks. We're going to look at how these building blocks form molecules. We're going to look at the biochemistry of membranes. And we're going to look at how everything comes together to form an architecture. We're going to look at some reactions. So we're going to do a little bit of math in here, not too much. And we're going to do some acid-base stuff, thermodynamics, redox reactions, electron transport, just a tiny bit of that. And protein conformation, we're really going to look into that as well. Why do proteins form the way they do? Learning objectives here is some of the things I would expect people to be able to do when you leave this course after 10 weeks. So you should know about basic cellular structure, biological molecules, do pH calculations, so on and so forth. I'm not going to read off this whole list for you. All right. So like I said at the beginning, the best way to contact me is through email. And you must do this through your Jaguar account, your student email. Us professors, we have been told, like, don't really respond to outside emails, even from a student. So make sure when emailing me, don't use like Gmail or anything. Use your student account. If your email is not working, I cannot help you with that at all. Make sure you contact the IT help desk if here, or their information is here. And if you don't hear back within 48 hours, just contact them again. They're usually quite busy the first week of a semester. So conduct and behavior. So basically, even though we're online still, which is hopefully the last semester online, knock on wood, I still expect people to be professional, no harassment, don't spam chat, all that. If you do have a question though, please don't hesitate to ask. And again, if you don't want to know, let the whole class know that you're asking a question, feel free to just send me a private message. That's totally fine. So exams will also be online. And I ask when you do an exam, for right now, we're not going to allow you to have phones, not going to allow you to have tablets, anything like that. At this point, I might change my mind moving forward. But right now, I'm going to say like no phones, no tablets. This was a really big problem during last semester where, everybody, it seems, was going to Chegg. And yes, professors know Chegg and professors know how to use Google. So it's not hard to figure out if people are cheating. So if you are caught using outside resources while taking one of my exams, I had to do this a lot last semester. This is what's going to happen. We're going to fill out a report. That report goes to the dean, goes to the dean of students. All that goes to my bosses to say that basically you cheated. I'll provide the evidence and then you'll get a 0 on the exam. And a 0 on one exam, it's going to be very hard to pass because since we have such a condensed schedule, I'm not going to be dropping any tests. But we'll get to that when it comes to grading in just a second. So let's get to grading then, probably your most important thing. So we have some different categories that will figure out your grade. First, exams. Looks like we have a question here. Yes.
Hi, Professor. I had a question about the exams. Is it going to be like through a lockdown browser or anything?
Probably. I haven't 100% decided which route I'm going to go with that. I tried a couple different things last semester, but I will let you know like at least a week in advance, like exactly what we're going to do for the tests. If there's going to be a lockdown browser, if I'm going to meet you on Zoom, I'm going to weigh the pros and cons of that in my head and talk to some other people to see what worked best for them.
Okay. And are we going to have like 24 hours to take the exam, or are we just going to have the class time to do it?
So since this is a synchronous class, I will probably just have it open for a smaller amount of time. The way I'm thinking about this right now is that on exam day, I will probably open up at 9 a.m. and have it open maybe till like 11. Since, like I said, this is synchronous, so I expect people to be here on that Monday and Wednesday.
Okay, thank you.
Yep.
Oh, I'm sorry. One last thing. If for some reason something does come up with those exams, would we still be able to take it like a day earlier or something like that?
Yeah, you can definitely take the exams earlier. Like if you have a conflict that you know of, let me know and we can just do it a day earlier. If something happens on the day of the exam and you can't make it, what I generally do for that is I will just replace your score on the final exam and just say like whatever you get in the final exam, I'm just going to copy that for the exam that you missed. That being said, if you miss like 2 exams, that's gonna be hard.
Okay. So, okay. Thank you, Professor.
Yep. All right. So let's actually go to grading now. So you have 3 exams in this class, and we're looking at 19% each. Final exam, cumulative, 25%. And then we have 3 other categories that are almost the total of 1 exam. And I look at these 3 categories as the way for you to really inflate your grade, in that I don't think these three things are hard. The homework, the reading guides, attendance, participation. I'm really rewarding you for being a student and having showing effort, right? So homework, what's the homework? So after every lecture, I'm gonna post some homework questions on Wileyplus. And like I said, I will show you where that is on Blackboard in a minute. Oh, well, once we get to the Blackboard section, I should say. And when I assign this homework, you'll have a week to do it. So let me see if my pen's working. Yeah, 1 week to do it, right? So after today, you'll have a homework assignment going over the lecture material we cover. That will be due in a week. When we meet for Wednesday, we'll have another assignment that I'll post after we're done. That'll be due next week, Wednesday. For these homework assignments, you have 3 attempts on every single question, and you do not get penalized for, or you don't get penalized much, I don't think, for getting a question wrong. I think it's currently set up that you don't get penalized at all if you like do 1 attempt and you get it wrong. I think you are perfectly fine. So I'm really, and these homeworks are just going to be trying to reinforce what we learned. So it shouldn't be anything new. It should just be review of what we did. So hopefully that's a way to boost your score. See what the chat's going on. Would it be possible for me to do the exam after 4? So like I said, this is supposed to be a synchronous class. So you're supposed to be here Monday, Wednesday, 8 to 10. So that's something we're going to have to work on then, because signing up for this class is supposed to be synchronous. So I expect everybody to be here. And the exam is really going to be open during that time, right? So that is something that we could possibly discuss over email. All right. So that's homework. Reading guides. So let's talk about this, and I'll talk about this again when we look on Blackboard. But before every class, except today, I will have a series of videos that I want you to watch. And these videos are basically material we're going to cover in the class for that day. So I don't want my students to come into a classroom cold. I want you to actually have an idea of what we're talking about for that day. I think that just really helps you learn better, because the first time you hear this information, it's going to be on a video. Then you're going to come to class and hear that information again. That's just going to strengthen your memory about that topic. While watching these videos, I will have a series of questions for you that you are to fill out. So make sure you fill those out while watching the videos, or you could also read the book and fill these out as well. I just figured students like videos more than reading at this point of society. So I made all those videos for you. And so you need to fill out all these questions and then turn them in before the start of class. And the way that it works is that since this is the first time you would be looking at this information, I'm not going to really grade for correctness. I'm going to grade for completion. So you get full points if completed on time. So, and I'll show you where this is when we get to Blackboard. But yeah, those reading guides, 6%, that should be an easy 6% of your grade. And attendance and participation is another 6% of your grade. Like I said, this is a synchronous class. So I do expect you to be here. We will also have questions during our lecture, and I expect people to participate in answering those questions. And I might call on you randomly to answer one of those questions, to see if you're engaged, see if you get it. So as long as you show up, and as long as you are participating, you get full points there. And the way I'm going to see if you're here is that I'm just randomly going to be taking screenshots of the people in chat. And if you're in there, you get points for attendance. All right. So just make sure you're using the same name on Blackboard as you are for chat, as that's how I'm going to be determining if you're here, which reminds me, this is a good time to take that screenshot. to make sure we're all here. No worries. Haven't missed anything yet. All right. Any questions about grading? And the letter grades are A, B, C, D, E, because we don't do pluses and minuses here. Oh, and here in bold, little to no opportunities for extra credit. This usually isn't a problem until the last week of class, where people are seeing their grades and then they message me of, hey, can I get extra credit? The answer is going to be no. I don't really do extra. There might be some time for extra credit during the class, but it's going to be rare. So any questions so far? All right. All right. So exams, when you get your exams back, when I make them available, feel free to message me if you don't understand why something was graded the way it was graded, or you think, hey, I should have got more points for this. I have no problems having those discussions at all. I welcome those because it actually means that you're thinking about things. So that's totally fine. Reading guides, those will be due at 8 a.m. Monday, Wednesday. So just make sure you do those. Exam dates, here they are. This is what I'm planning. So our first exam is 2.5 weeks from now. Since this is 10 weeks, it's going to be a very condensed schedule. Final exam, I know the dates between when we can have the final exam. I don't actually know the date of the final exam because they never release those except like 2 weeks before final exams start. So when I know, I will let you know. The rest of this stuff is going to be a lot of boilerplate stuff, statements you're going to find on every single syllabus here at Texas A&M San Antonio. So I will just briefly touch on this, and you have a copy of this. It's on Blackboard, so feel free to read any section that really pertains to you. But financial aid, Office of Financial Aid will deal with that. Disability services, if you need extra time, like on exams or anything like that, I cannot do that. You will need to contact Disability Support Services, DSS, for that. I have no control over that. So please do not ask me for extra time on exams. You will have to contact DSS and they will talk to me as an intermediary. What type of questions will appear on the exam? Matching, fill in the blank, multiple choice. Usually my exams, it's probably more short answer than most of other professors, but I really like short answer, some multiple choice, some select all that are applicable. So for that, I'll give you a list. And for those, if you answer the wrong question, I will take points away, but you can't get a negative score. So it's kind of like the SATs, except, your score for that question will never go below 0. I don't really do matching all that much. You might have 1 or 2 matching throughout the whole semester, and fill in the blanks also going to be rare. Short answer and multiple choices, mainly where you're going to get with a sprinkling of true false matching and fill in the blank, just rarely. So academic dishonesty, I kind of covered this, but this was a big problem for everybody that I've talked to last semester. So I just want to remind you that it's easy not to cheat when the class is easy, right? So it's really a measure of your character of what do you do when the class gets hard, right? Do you stick to those principles of I'm not going to cheat, or do you say, well, I'm going to cheat and I'm going to risk it? right? Because I don't care. Or actually, I care about the grade and that's it. So if you are caught cheating, like I said, I have zero tolerance for that. And I really have zero tolerance for when you're caught cheating and then you lie to my face, which happens in 90% of the cases. It's not fun. I didn't get into this job to be a private investigator and try to catch you in a lie, right? So if you are caught cheating, like I said, we have a meeting. In that meeting, I provide all the evidence. And then what's going to happen is that you're probably going to make some excuse about why that evidence isn't correct. But if you don't, we fill out a form together, and that form goes to all the higher ups. If you feel that what I bring forth is not true, you do have some recourse. And what will happen then is that if you feel like I accuse you of cheating and I bring forth the evidence and you say, nah, that's I didn't cheat, you have the wrong person, what will happen is that we will go and have a meeting with my dean. The three of us will sit together and have a chat. And if there's still no resolution after that, what will happen is that we will go to a committee, an academic dishonesty committee, and they will make the final call based on all the evidence presented. Their ruling cannot be overturned. So that would be the process. Every time I brought this forward last semester, it never got to my dean. Usually when I brought up the form, people signed it. So let's just have it this semester so I don't have to do that form. It's part of my job I absolutely hate. So just keep in mind, it's easy not to cheat when it's easy. It's really a measure of your character to not cheat when things get hard. All right, so veterans, active duty, we welcome. This is a big military campus. Feel free to contact the Office of Military Affairs for any questions. 6-drop rule. If you would like to drop, please do it before the automatic W date. Generally, after that date, unless I talk to you previously or there are some like very special circumstances, I generally don't give W's after that date, right? So if you are doing poorly in the class and you contact me 1 week before the final exam and say, like, I'd like to drop, can I get a W? And you're getting like a 50% in the class, I generally don't allow that, because the cutoff date's there for a reason. So just be aware of that date, which I have at the bottom. Writing Center, we're not really going to do essays this semester, but the Writing Center is a great place for it. If you do have writing, I've sat in on some Writing Center appointments there. They do great work. Please see them if you have to do any writing at all. Academic Learning Center has tutoring. I don't know if they have a biochemistry tutor right now. They used to, but I don't know about right now. But you do have me as well, the person teaching this class and who has taught this class for like, I think this is my 7th time teaching this Biochemistry 1 class. So I'm a pretty good tutor too. I write the test, so I know what's going to be on the test. So feel free to contact me for tutoring as well. Counseling. As a student here, you do have free counseling. So feel free to contact them on this number here. However, if it is an emergency, there is a 24-hour hotline number here as well that you should contact instead. COVID-19. So we're not meeting on campus for this class. So if this is your only class, it's not that big of a deal. And we are winding down in the pandemic. But since half the population of America refuses to get vaccines, we're going to still be dealing with it for a while. So just be aware that if you're feeling ill, don't come to campus. Even if you're vaccinated, there's still a 5% chance, give or take, that you can still catch COVID-19. So you're not immune, you're just very unlikely to get it. But 5% is not nothing. So just be aware of that. So if you do have like headaches, chills, any of these, best to stay home. And hopefully during this course, we will have some examples. Well, we're definitely, if we get time to it, we're definitely going to go over how vaccines are made, all the different trials they usually have to go through. We might have some topics that talk about COVID-19 specifically when we talk about membranes and proteins. So we're going to talk a little bit about COVID-19 this semester. But yeah, if you're not feeling well, don't go to campus. So that's all there. So key dates. So the 7th is our first day of class. July 16th, next month, is that big day where you need to drop to get a W. And let me just check when our tests are. Yep. So that should be right after exam 2. So you would have 2 exams in and you'll have a good idea of how you're doing in the class. If after grading exam 2, it's like the 17th or 18th, and you thought about it for a couple of days and you're thinking, I don't think I'm gonna pass, just contact me. And just a couple of days after that date, I would give you a W still. But if it's like after test 3, and you don't do well again, you're unlikely to get that automatic W. So just keep that in mind. The 29th is the last day to drop the course at all. The 10th is our last day of class, and I'll let you know when the final exam is. These dates can be found if you Google academic calendar. So it would be, if you're interested, Texas and San Antonio academic calendar. That's where I'm getting these dates from. So if you're ever curious about that. And that is the syllabus. Now, before I move on to Blackboard, any questions about the syllabus that people might have? All right, so just give me a second here. Get my pen off, stop sharing. Let me make sure the Blackboard is good to go. And then I'll share Blackboard. All right, so since this course is online, our Blackboard site is going to be used a lot. I just opened here in our announcement section, right? Any announcements that I make will appear here, and I will also try to remember to click send to email. So you should get that popped up in the email. Syllabus has a syllabus. It's what you would expect. Course content. This is one of the big things for us. And I forgot this thing is hidden. So let me unhide that. All right, so folders. There's going to be a lot of folders under course content. First, let's look at this folder called Test 1. Test 1 will have all the PowerPoints we go over that I will ask for on Test 1, right? So the information in the Test 1 folder is what I'm going to test you over for Test 1. And I call them problem sets. They're really PowerPoints, but inside the PowerPoints, I have a bunch of problems that I ask during class. And these are to test your mastery of the material. And also as a good way to show you, hey, I'm asking you a question in class. there's a good chance I might ask you a related question, or rarely I will just ask the same exact question word for word on the test. So these are a good way, those questions in there are a good way to really test your understanding. For every single class, I will have a different problem set. Right now I have this week's up. As the semester goes forward, more and more will pop up in test 1. And then we'll have a folder called test 2. We'll have a folder called test 3. I noticed you put the reading guide on the academic calendar. Oh, you mean on Blackboard, I think. I can't do that with homework. So our homework is run through a different program. So those, you're just gonna have to remember they're due Monday, Wednesday, 8 a.m. I'll show you where to access it, but I don't think I can link that to your calendar the way that the homework is run through a different program. Right, so that's the folder called test 1. And I will have another folder that says test 1 answers. So for all these problems that we go in class, I have an answer key that I'll make, and I'll just post that answer key online, right? I will most also likely put study guides up. These study guides will be found in the same folder. So like problem set 1, you might see a file called study guide 1 here throughout the week as another way to just test yourself. And I will also put up in the test 1 with answer folders, the answer to all those study guides. I will say the best way to study for this class is to do all the questions we have in the PowerPoint, do the questions in the study guide, and don't look at the answers until you answered everything, right? If you look at answers and just try to memorize the answers, you probably won't do too well because like I'm going to ask questions on the topic, but it's not going to be word for word the same question, right? So if you just memorize the answer, I might ask like a question on the topic, but in a different way. For example, from general chemistry, what happens when a positive and negative ion come together? They attract. A way I might ask that on a test is I might say, okay, what happens when a negative ion and a negative ion comes together? Well, they repel. And you would understand that if you like thought about the question, what happens when positive and negative come together? If you just memorize the answer, you might put, oh, here's a question about 2 ions coming together, so they must attract, and you would get it wrong. So that's just kind of an example of what I mean by the questions I give on the study guides and in class. When it comes to the test, I will ask questions on those topics, but they're not going to be word for word the same. So make sure you understand the topic, understand why you answered the way you did answer, and then you should do fine. All right, so those are the folders called tests. You can also see that every single date, I have already created a folder. So let's go into the June 9th folder. Here you can see I have a bunch of videos. I also have the PowerPoints for these videos. One thing that I will ask you to do as your way of rewarding you for being prepared for class is, like I said, I want to have you watch these videos, and you will have this reading guide. So open up this reading guide when you're watching the videos, fill it out. When you're done, put it in this Turnitin. So make sure you do that before 8 a.m. Make sure you get a confirmation from Turnitin in your email that it was accepted. This always happens where somebody thought they turned something in and it didn't go through. I can be lenient about that when it happens just a few times. But if it's becoming a constant thing, then we'll have to work something else out. But yeah, here's the reading guides. Here's what you're going to turn it in. And for every single date, I already have this up. So if you'd like to get ahead of things a little bit, feel free to just work through these folders. If somebody was really gung-ho about biochemistry, you could probably get all this done like by next week. I'm not asking you to. I think that would be a little too much, but just make sure you're working through these and you get the reading guides done before 8 a.m. on the date it says. So reading guide number 1 for Wednesday, June 9th, that is due 8 a.m. June 9th. So make sure you do that. Also, under course contents, this is where your tests will appear, right? So when we have tests in class, there will be a new link that appears that is just simply called like test 1. And you click on that, and that's where all your tests will be. It will not be under exam and quizzes, so let me hide that link. All right, great. So what I am going to do is, that's not where it is I'm going to set up the grading scheme later today. And in that grading scheme, there's going to be one column that's incredibly important. It's called grade in class. It's not there yet because I haven't made it. But by the end of today, probably this afternoon, you're going to see a column that says grade in class. This is what you expect. This is your current grade in class. So this will update every single time I grade something, right? So you can have a real live and look of what your current grade is. Keep in mind that this can fluctuate a lot before the exams are graded because there's not much going into your grade. And then at the end of class, when we get near the final, reading guides and homeworks won't be changing this all that much anymore because we have the bulk of material in. But yeah, if you're ever wondering what your current grade is, that's where it will be at. One other thing about grades, let me go there. Go to the full grading center. Wait. So reading guides. You see I have a column for each reading guide. These are the turn it in columns. I will not put a grade there. So do not be shocked when, the end of the semester is coming and you see all these reading grades being labeled as ungraded. I will create a new column for every single reading guide and grade it in that new column. I need to do that because of that grading class. For it to work, I just need to do it in a different way. So just keep in mind, these reading guides, they will say ungraded, but at the very end, I will create a new column where they are graded correctly. So that's great. The next big thing that's super important for you, tools. And I didn't realize I had that hidden, but it should be available now, tools. Under tools, the only link that you care about right now is called Wileyplus. So I'm going to say that again. Tools, Wileyplus. When you click on Wileyplus, you're not going to see everything I see. Yours is going to be much shorter. And let me go student preview, actually. So I go to tools. I go to Wileyplus. Yep. So here's what you see. Two things are going to be important for you. Wileyplus assignments. Homework is here. So this is where your homework's going to show up. This is why I can't link it to like the academic calendar, or at least I've never found a way, because it's in this separate program that's under tools. The other thing that's important for you is Wileyplus Read, Study, Practice. This is the ebook. Although I have videos made for everything we're going to cover already, I still think it is helpful as another way to reinforce this material to read the ebook. The ebook has more examples than I give during class, so it might be useful to see material presented in a slightly different way. Do keep in mind that the ebook will have information that I don't cover in class. And if it's information I don't cover in class, I'm not going to test you on it because that's not fair, right? If we don't go over it, I'm not going to test you on it. So just be aware, if you read something in the ebook and you're like, we never talked about this, don't worry about it. You're not going to be tested on it. Also, when talking about the ebook and all the study guides that I'm going to be posting, just be aware, and you might have heard this before, that for every hour you spend in class, you should be spending 2 to 3 hours outside of class studying. And every time people say this, I know I did this as a college student, I would laugh at that and be like, yeah, I'm not going to spend that much time outside of class. Then organic chemistry hit me in the face and I realized, oh, for these science classes, you do need to spend that much time. So as everybody has made it through organic chemistry, hopefully that kind of statement is not all that surprising to you at this point in your career. Usually when you get to me in biochemistry, you're pretty strong students up and down. So I don't really need to reinforce that all that much or remind you of it, but just be aware, this is very dense. We're going to go quite quickly, especially these the first chapter. We cover a lot of material in a very short amount of time. Just be aware that you're going to have to be putting in this extra work to really do well in this class. Do you believe it would be more beneficial to read the textbook before lecture or after? I think I would probably be in the camp of before, in that when you read it before, I wouldn't struggle that much about trying to nail every single concept down. That is, if you read something and you can't make like sense of it, I wouldn't spend like 15 minutes of my time trying to understand that like one concept. What is really beneficial for the before class activities is that when I say something in class, it's not like 100% new. You kind of have like, oh, okay, I kind of remember reading that in the textbook, or I remember listening to that in the videos. And then when we're going over through it in class, after you hear my explanation, if it still doesn't make sense, that's the time to really like ask the question of like, so this topic, I looked it over in the textbook, I watched the video, I didn't understand it. The explanation you gave, I still don't understand it. Could you help me with that? And then I'll be happy to do that. And then afterwards, what would be great is if you go back to that section of the textbook that you're like, I didn't know what was going on there, and reread it and see if it makes sense now. Because learning, well, so let me just say a quick thing about learning, kind of off tangent. I'm a firm believer that being intelligent isn't just a like genetic thing. Sure, how quickly someone learns something That is, has some genetic components. People are different, but I'm a very firm believer that anybody can do this stuff by trying hard, right? So don't ever feel like, oh, I'm not smart enough to get this stuff, or I will never be smart enough to get this stuff. And don't try to compare yourself to me. Like I said, this is like the 7th time I've taught this class, so it'd be kind of shocking if I didn't know this stuff by now, because learning is just repetition, right? So the more reps you put in, the better you're going to learn this. So that's why it would be great if you read the textbook before, watch the videos before, come to lecture, be engaged, and ask questions, answer questions. Then afterwards, go back, review your notes, review the textbook. And if you just keep hammering the same information like that, your neurons are just going to be super strong and you'll remember that stuff. So I'm also a big believer of flashcards. For the last 2 years, I've been learning a foreign language, and I now have 7,500 flashcards. And I have a flashcard app that costs me 99 cents. And every single day, I spend about 40 minutes a day just reviewing flashcards and making new flashcards to learn that language. So biochemistry or any other class is going to be the same. Repetition, repetition, repetition. Space out your learning. The worst thing you can do for an exam is cram the day before. The best thing you can do for exam is study every single day consistently. And then right before the exam, that's going to be like, okay, is there any topic that I really don't understand that I can really devote my time to? And the other topics you just should have down better. So while you're still in the first week of the honeymoon phase of going back to class, where everybody's actually gung-ho and shows up and does assignments, before that spirit drops off, I really advise you to pick a time when you're going to focus on studying for your classes. During that time, I really suggest that you don't have the TV on. You don't have your phone on, maybe except for a timer. You don't go to different websites. You just focus. You do that for maybe an hour a day, maybe 5 days a week, 6 days a week. You will be perfectly splendid for this class or any class that you really have to take. That would be my advice. And that was kind of an off tangent answer to that. But yeah, that would be my advice for studying. All right. And I think that is, let me clear my writing. That is the main points of the Blackboard site that you need to look at. Everything else should not be applicable to you. Don't try to send me an email through Blackboard. It seems like there's a 50% chance that if you send me an email through Blackboard, it's just lost and I never see it. I don't know where it goes. It just goes into the internet ether. So yeah, just send me an email, candrews@tamusa.edu. I do have a second email, which is casey.andrews@tamusa.edu. For some reason, I find C. Andrews to be better. Sometimes when people use the other one, again, it gets lost. I don't know how, but it does. So before we jump in to actually get into biochemistry, any questions about the class itself, grading policy, anything non-material wise? On the syllabus, I do have times for office hours. And what I might do for that is I will probably just be in the room we're in right now, working on other things so people can pop by. But I'm basically open anytime for a meeting. So if you want to meet, just send me an email and that's no problem. All right, if there's not, let's jump into it. And reminder, what we're going to look at is the PowerPoint problem set 1, June 7th, again, under course content, test 1. So let me stop sharing this. Let me share the PowerPoint. And we are going to start Biochemistry 1 proper now. So before we get in, be aware that I don't know why, but my computer cuts off the screen below this arrow. Like I can draw below there, but you can't see it. So there could be some times on the PowerPoints we go over where the bottom's cut off. I will do my best to rewrite that information. Just be aware that like on the actual PowerPoint that you can download and follow along or print out, the information should be there. All right, so we're going to start with 1.1 to begin with. And our first chapter of biochemistry is all the things that you should have learned in organic chemistry, biology, and general chemistry in the span of like 1.5 class periods. So this is a lot of review. It's also a lot of material that I'm not going to spend too much time over unless there are specific questions. But since biochemistry is the marriage of biology and chemistry, we have to know information from both of those classes. And we're just going to start with a question. And I can open this up for anyone if you want to answer in the chat. But if you look at the human body, what element in percent dry weight would you think is the most abundant? And B, if you had to pick one molecule in the human body that appears the most, what do you think that molecule is? What are we made out of? Carbon, that is correct for A. That's the reason why organic chemistry is a prereq for this class. Every single molecule pretty much we look at this semester is going to be mainly made of carbon. Remember, carbon has 4 bonds. It can do some funny stuff like double bonds. It can also do triple bonds. But carbon, if we look at dry weight, dry means without water, is our most abundant element. And yes, what molecule is the most abundant in the human body? H2O. We are roughly 70% water, right? So we're chapter 2, we're going to look at the chemistry of water. Life happens because of the interesting / weird properties of water, right? All of our proteins work because they're surrounded by water for the most part. So we're going to get into that. So those are the things we're really going to be focusing on: carbon chemistry and why water makes such a big influence on living creatures. So where did all these chemicals come from? Where did carbon come, or where did the amino acids come from? Where did DNA come from? And people have been thinking about this for a very long time. How did life just spring up? And scientists did a couple of different experiments to try and look at this. Operating in Haldane in the 20s, Miller and Yuri in 1953. And the image I have over here is the Miller and Yuri experiment, which is a very famous classical experiment in biochemistry. So let me just take a minute and explain what the Miller and Yuri experiment is. So they have this closed, more or less closed system, right? In this system, they have gases. And these gases are what we think early Earth's atmosphere was made out of. So methane, ammonia, hydrogen gas, a little bit of water vapor, right? And we have the ocean here made out of water that will be boiling. So it's very hot. That's going to send water vapor to our chamber. And we have electricity here. So we have electrodes, and they're going to be giving sparks in this chamber. Think lightning, right? Lightning hitting sparks. And what Miller and Yuri saw is that if you have these gases inside of a container and you hit it with lightning, you make amino acids. So like glycine spontaneously creates under these environments. Alanine spontaneously creates under these environments. So we have been able to just randomly make the amino acids found in our body by doing a simple experiment like this. So that's one idea. Other scientists think that the biomolecules found in life most likely came from thermal vents. Thermal vents are these very hot vents, mostly under the ocean, 400 degrees Celsius, so incredibly hot. And at this temperature, you can have some really interesting chemistry happen. These also emit metal sulfides, which can be used as catalysts. And so these scientists think, maybe the amino acids, maybe RNA was able to be created under these very high heat, high pressure conditions. Of course, we'll probably never know this because no one has a time machine. Plus, traveling back in time is basically impossible anyway. Sorry, sci-fi fans. Due to law of thermodynamics, you can't travel back in time. You can travel forward in time, but not back. So we'll probably never know the answer to this, but those are just some experiments looking at these chemicals. So what are we going to look at in terms of molecules for biochemistry? And biochemistry, unfortunately, there are some memorization that we'll have to do. And here are some common functional groups, name and structure. So table 1.2 and table 1.2 continued just has names and structures. And unfortunately, I'm going to ask you to know names and structures to begin with. So like I said, this is one of our big memorization things we have to do at the very start of the semester. And the reason I'm asking you to do this is that when we get into biochemistry proper, like in chapter 3, 4, 5, moving forward, these things pop up all the time. Disulfides, amides, carboxylic acids, all that stuff. And so it's really helpful in our discussion if I can say the carboxylic acid is doing a is doing a cyclization reaction right here. And it would be great if I didn't have to sit down and point out where the carboxylic acid was or explain what that is. So this is part of our vocab. So make sure you look at these. What I might actually do is, since I was talking about that flashcards I have for learning my language, that's all on my phone. And I got pretty good at making those like decks. So I might just turn these into flashcards and provide the link. The flashcard app I use, though, might cost you 99 cents. So I know, big money. But yeah, I can do that for you so you can study with those. But just make sure you know those. When it comes to the test, most likely how I'm going to ask these is I might give you a name and then multiple choice of different structures, and you just have to pick the right structure. Or I'll give you a structure, multiple choice of the names. I'm not going to ask you to draw anything because drawing online is a big pain. And another reason why I have you to understand this, if you understand the chemistry of these functional groups, you know how proteins are going to behave, right? Proteins and other biological molecules are made out of these functional groups. So they behave like their functional groups. So it's just a way to simplify our understanding. If you know how it works on the small scale, you can understand how it works on the large scale. So that's our first memorization topic. So here's just a little bit of review for us. See if you remember organic chemistry. 1, 2, 3, 4, or 5. Which one of these is a carboxylic group? So we do have some answers. 5, 5, 5. That's correct. Classic carboxylic acid, C double bond O, OH. Usually in biochemistry, we're going to see it like this. Usually in biochemistry, that hydrogen is not there because the inside of a cell is roughly a pH of 7. But yeah, these are carboxylic groups. They appear all the time in biochemistry. Negatively charged, and they can also be involved in hydrogen bonding as well. All right. So one of the big topics that we're going to hammer on in biochemistry is this idea of polymers and monomers. And in fact, 80% of the class, maybe not that high, maybe like 70% of the class, is just looking at these monomers and looking at what type of polymers they make. And we are concerned about 4 different molecules. We are concerned about proteins, nucleic acids, polysaccharides, also called sugars, SUGARs, and lipids. So we're going to look at all 4 of these molecules in depth. Monomers are made out of amino acids, nucleotides, monosaccharides, and fatty acids. So these monomers connect together to make polymers, and these polymers have both covalent and non-covalent bonds. So just in case it's been a while for you, let's say I have this molecule and I have shared water. So covalent bond is like the actual line there, right? It's a line between bonds. They are sharing electrons. A non-covalent bond would be an interaction between this oxygen and hydrogen. And what I'm actually showing is a hydrogen bond. Some hydrogen bond. Non-covalent bonds, they are most likely what's called transient, in that they can break. Not always, though. We're going to look at cases where non-covalent bonds hold together pretty well. But covalent bonds, like, never break, almost. Almost never break. You need a lot of energy to break a covalent bond. You don't need that much energy to break a non-covalent bond. Like a covalent bond is roughly like 20 times stronger than a non-covalent bond at least. So it takes a lot of energy to break this. Not too much energy to break this. But our polymers are made out of both, as shown here. Our lines here are covalent bonds. How these are interacting across, these are non-covalent bonds, non-covalent interactions. And yeah, we're just going to look at how these polymers are made in this class. But before we go into that, let's do a little review and see if we can just recognize the different molecules. And let me pull up, let's see how easy it is to get a poll here. Right, these polls are not easy to make. So next time I'm going to have all these polls already made. But yeah, so what I'm showing here is a molecule. What type of molecule do you think I'm showing? Do you think I'm showing a lipid? Do you think I'm showing a carbohydrate? Do you think I'm showing an amino acid? Or do you think I'm showing a nucleotide? Oh, take about 30 seconds or so. See if you can answer that. Yeah, very good. So got an answer from a few of you. This is an amino acid. So how can we tell what an amino acid is? The box I just drew is how we're going to tell an amino acid is an amino acid. Every amino acid, and we're going to go into this in depth, always has the same chemical structure. It goes NH3, or this can also be NH2, or it can also be NH, to carbon. And off this carbon, we have an R group. And this R is just like anything. Then we have a carboxylic acid, or it could be C double bond O, or for some reason, every time you look at amino acids on the internet, and I don't understand why they do this, they put C double bond OH. So that's internet. That's also stupid because inside the cell, amino acids will never have that hydrogen. Actually, the same thing with the NH2. That's the internet. That's also stupid. I'm not saying you're stupid if you write that. I'm just saying it's stupid that the internet puts that up as your first result when you look at amino acids, because it's always going to be NH3, C double bond O. We'll talk about why that is when we get to the amino acid section. But yeah, when trying to determine what an amino acid is, look for this backbone. All right, how about this? What type of molecule are we looking here? And so I'm gonna do a random call here. Let's see, who have we not heard from today? Let's go, eeny, meeny, miny, mo, pick a tiger by his toe. If he hollers, let him go, eeny, meeny, miny, mo, and you are it. Let's go, Brandon. I have not heard from you today. Brandon. And feel free to say this in private chat, or you can say it out loud. And the rest of you, even though I didn't call you, please think about this. But what type of molecule are we looking here? Is this an amino acid? Is this a lipid? Is this a carbohydrate? Or is this a nucleotide? No, it is not that. Good try though. So let's take a look at what this is. This is a lipid. So usually when I ask this question, people are usually torn between two things. Is it a lipid or is it a carbohydrate? Let's go over the differences now. So carbohydrate, let's break down that word. Carbo, carbon. Hydrate, water. So when looking at a carbohydrate, you're looking at carbons that have a, oh, that was a very bad drawing, has a bunch of OHs on it. Carbohydrate. For a lipid, what you're looking for a lipid is a long chain of carbon and hydrogens, and usually a polar head group. So here we have an OH, but everything else is carbon. If this was a carbohydrate, we would just have like an OH on like everything. So that's how you can recognize a lipid versus a carbohydrate. That is another one of the class of molecules we're going to look at. Let's keep the good times going then. Here's another class of molecule. Hopefully you've seen this before because you've taken general biology. What are we looking at here? Is this a carbohydrate? Is this a lipid? Is this an amino acid? Or is this a nucleotide? Yes, very good. That's a nucleotide. Funny enough, it is made out of a carbohydrate. So here is your carbohydrate. But taken together, if you have a carbohydrate with a base and a phosphate, this is a nucleotide. So that's what you're looking for there. Carbohydrate, base, phosphates. And we'll get into the structure of nucleotides probably like in a month from now, I think, if I remember my timing. How about this? Here's a different way. We have not looked at molecules before. So let me tell you what you're looking at. This is an image of a molecule that's showing polarity, right? Kind of showing polarity. The key codes is that red is oxygen. Oxygen is usually negative. Blue is nitrogen. Nitrogen is usually positive. Here, white is hydrogen. Hydrogen is neutral to positive. Green is carbon. Carbon is neutral. So let's see here. Usually I have a list of people, but I did not make that. So let's go. How about Ariel? Ariel, what molecule do you think we are looking at based on this image? And this is probably one of the harder questions I ask when it comes to this, because we're looking at a molecule in a different way than we looked at before. Everyone else, see if you can also decide what this molecule is. Correct. That is an amino acid. All right. So how do I know, or how do you know rather, this is an amino acid? Carbon, oxygen, oxygen, carbon, hydrogen, nitrogen, hydrogen, hydrogen, hydrogen. Here's our backbone that we talked about. I'm just showing you it in a different way. And then we have a bunch of other molecules off the center carbon. So remember, with the amino acid, always look for that backbone, C double bond O, another oxygen, carbon, and H3. So very good. And lastly, we have this molecule. I already talked about it, so we'll just go ahead and say that is our carbohydrate, right? Carbons, oxygens. So those are going to be the 4 main classes of molecules we look at this semester. This is mainly an introduction to what they look like. We're going to go into depth on each one of these in due time, look at all of their chemistry and how they react. And that brings us, so that was all of 1.1, your basics of orgo/biology. Now we're going to go more into molecular cell biology, just a little bit, cellular architecture. If you never taken cell molecular biology yet, don't worry. This is, I say it's cell molecular biology, but it's mainly Gen Bio again. Not all, 90%, 95% of the chemistry we look at this semester takes place inside the cell. And if it doesn't take place inside the cell, it takes place in the bloodstream. So it's very important to have an understanding of the cell, both prokaryote and eukaryote, right? So we care about eukaryotes because we're human, and so we want to understand the biochemistry of ourselves. But when studying biochemistry, usually a lot of the studying in labs is done on prokaryotes because they're easier systems. So that's why it's important to know both prokaryotes and eukaryotes. And inside the cell, when it comes to definitely eukaryotes, but also prokaryotes itself, why even create a cell, right? What is the advantages of having a cell? And then taking that one step further, why do you think eukaryotes took that idea of compartmentalization and went crazy with it? Why would you ever compartmentalize? Why don't living organisms, why do we need cells? What are any advantages to it? I'll have this question for anyone who would like to put forth an answer. Why do you think we want to compartmentalize? Both prokaryotes in that they have a cell themselves, and eukaryotes, which we have organelles. Protection, that is, yeah, that's good. Protection, and let me just kind of expand on that deal. Not only protection, but you get to choose which molecules enter. For not always, but somewhat. So if there's a molecule that's poisonous, sometimes as a cell, you can choose not to intake that. Like I said, some poisons just get in no matter what you do. But yeah, protection. Easier to maintain. I'm gonna kind of maintenance. That kind of goes along with that, in that if you are contained in a cell, you can better get rid of your toxins or keep good stuff. If you don't have a cell, it's just all out there, and maybe that ATP you wanted just floated away. Bye-bye. And now it's gone. Ability to have metabolic pathways. Yeah, that's kind of going along with maintenance as well. Because when doing metabolism, the main way you make ATP is the proton pump, where you have one area that's high in hydrogen and another area that's low in hydrogen. And by letting that hydrogen flow through ATP synthase, you make ATP. Also, with metabolism, this is more of a eukaryotic thing, is that with our different organelles, and that's also here, we can have efficiency. Because with organelles, we can say, you know, I want glycolysis to run in the cytoplasm. And I want the gluconeogenesis that is making glucose, I want that to run in this different organelle here, the mitochondria, right? So by having these compartments, you're making sure that these two processes that are opposite of each other, you're not just doing a futile cycle, right? And allows for communication among organelles. Communication. So by having compartmentalizations, you can send specific messages to different organelles, and then you can turn those organelles on and off. Prokaryotes, since they don't really have organelles, well, they don't have organelles. They don't have this, but you could still turn on and off specific proteins. So yeah, very good. I would just say, like, kind of what everybody was hitting on there was this idea of with compartmentalization. You can really be in charge of concentrations. So you can have a lot of ATP inside your cell. In one part of your cell, you can have a lot of glucose. In another part of your cell, you're going to have a high concentration of some other protein or some other macromolecule or biomolecule. If we didn't have compartmentalization, it would be very hard to do reactions because these metabolites would just keep floating away, right? So that's mainly why the cell membrane was created, or evolved rather. That's why eukaryotes took that a step further and just make organelles, this compartmentalization to keep everything separated, to keep our metabolites separated so we can do efficient chemistry. And we're just briefly going to look at the difference between prokaryotes and eukaryotes here. So on the left, I have some prokaryotes. On the right, I have a typical, I believe, an animal cell. Size-wise, the size of a prokaryote is, let's say that's a prokaryote. A eukaryote would look something like that, maybe even bigger. So eukaryotes are way bigger than prokaryotes. Big difference between prokaryotes and eukaryotes, organelles. So the eukaryote here has different organelles. The prokaryote doesn't. So that leads us to this question. Which of the following do eukaryote cells have that prokaryotes don't? DNA, cell membrane, nuclear membrane, ribosomes, or proteins? So what's the difference between the prokaryote and eukaryote? We have a vote for nuclear membranes. Do prokaryotes have DNA? We have a vote for cell membrane. We have a vote for a cell membrane. Do prokaryotes have ribosomes? So the answer is it is C, nuclear membranes. All right, so the cell membrane is being shown here, right? Every cell has a cell membrane. The cell membrane, also shown there, is what keeps everything inside. Anything inside the cell membranes, the cell. Anything outside is not the cell. So to be considered a cell, you need a cell membrane. The nuclear membrane is only found in eukaryotes. So that's the organelle. The nuclear membrane keeps all of the DNA in one spot for the eukaryote. The prokaryote doesn't have one of these. So if you look at a prokaryote, let's say this is the prokaryote, the DNA is just all over the place. It is just floating around in the cytoplasm at all times. We don't have that. We create our own membrane. Ribosomes make proteins, so that's found in everything. DNA, found in both prokaryotes and eukaryotes. That's your master system. Proteins do the work. Just some other examples that we're going to look at this semester. We'll look at the mitochondria. We're going to go in depth into the Golgi apparatus, talk a little bit about the lysosome. We're really going to cover cell membrane, so that's going to be the big one that we look at in depth. There's a lot of biochemistry with the cell membrane. Yep, so just make sure you remember that. Eukaryotes big, prokaryotes small. Eukaryotes, membranes, organelles, rather, prokaryotes, no organelles. All of their proteins are just in the cytoplasm or in the intermembrane space. All right, so let me just take a pause there to get a drink of water, but any questions about anything we've covered so far? And just as a reminder, these type of questions we're going around class, they're similar to what you might see on the test. And the first chapter of biochemistry is really just review. So any questions up to this point? Any clarifications? Not yet. All right. And so talking about biochemistry, talking about the cell, we look at the tree of life here. We can be broken into three major categories: eubacteria, archaea, eukaryota. The archaea and eubacteria, those are going to be your prokaryotes. The eukarya is going to be your eukaryotes. When you look at branches, those are common ancestors. So if you look, and we're going to talk about a little bit in this class how we can use proteins and DNA to make these type of charts. I always find it interesting when looking at this that animals, their closest ancestor is to a fungus. So out of all the eukaryotes, if you just had big broad classes like this, we're most like fungi. Archaea, really weird. I don't know if weird's the right word, but interesting, I guess. Archaea live in extremely inhospitable conditions. These are the microorganisms you will find in super salty lakes. These are the organisms you find by vents where it's like 400 degrees Celsius. So that's archaea. Eubacteria, that's going to be all your bacteria that you know and love, your E. coli going to be there. Those are all your prokaryotes. So that's the tree of life as we know it. Very, very zoomed out view of that. And when we talk about evolution, well, hopefully you already learned some about that in your class, your classes. So let's just think about evolution. And we have 5 questions here. So what I'd like everybody to do, and take a minute, and you don't need to put it into chat right now. Just take a minute and ask yourself for these 5 questions. Are these true and false based on how you think about evolution in a scientific sense? So take a minute, write these down. Well, not the questions, like your answer to these. yeah, so I'll give it a minute for people to do that, and yeah, take it from there. All right, so I think what I'm gonna do for this is I'm gonna call on individuals for each question. So A, let's go with, let me just make sure these people are here, I suppose. Okay, Jude, what do you think A is true or false, that evolution is not directed towards a specific goal? True, that is correct. Although humans like to think of themselves as the end-all be-all for evolution, we did it, we're the final form. Evolution itself as a scientific principle, there's like no idea in mind. It's adapting to new scenarios or different niches that you can take advantage of. Like for example, in the ocean, we have giant plastic garbage pits. We are now finding bacteria that can eat that plastic. They found a food source and now they're breaking it down and now they can multiply. So that's an idea of evolution there. That is true. 2, B. Let's go with Carolyn Cardoso. Evolution is ongoing and is constrained by its past. Do you think that is a true or false statement? True, that is correct, right? As long as there's pressure, evolution won't stop, but you can really only create things if you have some kind of template there to begin with. So for example, humans probably won't evolve to have jet propelled like wings because we don't have in our DNA how to create like combustion reactions. So that just won't spontaneously happen. But you know what does happen is people are born with like webbed feet and hands. So that's in our DNA because that's in the past where our common ancestor probably had like webbed appendages. And who knows, maybe in 3,000 years from now, this will just be an ocean planet. Maybe everybody have webbed hands and feet. Who knows? But yeah, true. C, evolution. Oh, let me just make this disclaimer. I know evolution can be a touchy subject for people, but this is a science class, so all I care is that you understand it. If you have personal beliefs that say, I don't believe evolution as presented here, fine, I don't really, that doesn't bother me. I do expect you to know the scientific basis of evolution because this is a science class. All right, so C, evolution requires some sloppiness for adaptation to changes. Let's go with Lydia Smith there. Do you think that's a true or false statement? So let me just write down what we have so far. True, true. True, question mark? Yes. Let me explain what I mean by sloppiness there. Mistakes. Evolution does not happen unless there are mistakes. You have a mutation in your DNA, right? If our molecular machinery worked 100% and never made a mistake, you would never evolve unless, you had some kind of like someone changed your DNA. Evolution only happens because your cells make mistakes. They make mistakes very rarely, but they do happen. These mistakes lead to change. And that leads us to D. Variation among individuals allow organisms to adapt. And who did I have for that? Olivia Rodriguez. Variation among individuals allow organisms to adapt. True. Yep. So if everybody was the same, evolution would be a lot slower. So you need the differences in individuals to allow for evolution to happen. And E, changes in the DNA of somatic cells is key to evolution. I'll answer that because that's the only one that's false, right? So somatic cells, remember you have two types of cells, somatic and germline. Germline is your sperm and eggs. Somatic is not your sperm and eggs. So it's only a change in these organisms that you can pass on. So a classic example that's really not this, but kind of the same idea. If you are like a bodybuilder and you get super buff, that does not mean your baby is going to come out super buff, right? Because you didn't change anything about your germline cells by working out. You're mainly changing your somatic cells for that. So yeah, only changes in your germline can be carried forward. So that's our little review on evolution. Questions so far about anything up to this point on our whirlwind tour of everything you should have learned in different classes? All right, if not, we will continue on here and get to everybody's least favorite review chapter, thermodynamics, aka math, aka why everything in the universe happens. Entropy, enthalpy, delta G. My favorite subject because when I went to grad school, I was a physical biochemist, which means I really did all the physics of biochemistry, which is thermodynamics, which is one of the things of physical biochemistry. So let's just talk about how physics relates to chemistry. Life is never at equilibrium. You never want to be at equilibrium. The only time your cell gets in equilibrium is when you die. So to make sure we're not at a state of equilibrium, you eat food. That food you use to make ATP. ATP is used to do things outside of equilibrium. So we're in a constant fight of returning to equilibrium as a living organism. We do that by transforming energy from our surroundings, eating food. First law of thermodynamics. You cannot create or destroy energy. You can only change forms. All the energy in the universe is constant. Doesn't increase, does not decrease. It just changes from energy to light or to work. And when we talk about physics, some definitions, The system is everything we're interested in. So reactants, products, solvents. The surroundings is everything else. So it's really simple. System, what you're looking at. Surroundings is not the system. Anything that is not the system is the surroundings. System plus surroundings equals universe. Everything is part of the universe. When looking at systems, you have three different types of systems. Isolated, which means you do not exchange anything. You have closed, which exchanges energy but not matter. And you have open, exchanges both energy and matter. Life, we are an open system. Not only do we exchange energy with our surroundings, we also exchange matter with our surroundings. We do that all the time. So that was the first law of thermodynamics in 2 minutes. Let's go to the second law of thermodynamics. That is randomness in the universe is always increasing. The technical term for randomness or disorder is called entropy, which I actually have down there. So every second of the day, the universe is becoming more and more random. We can look at this through, when we look at the universe, we see that it is moving apart from itself, is just spreading out. And that is a way to increase disorder. And I'm showing that example over here on the right. So we have gas in two containers. If we have the gas in just one container and we open up the bridge between these two containers, this is, the bridge is here is closed, this is open. Spontaneously, the gas will go from one container to another and spread out, because this increases randomness. This is more ordered. If all the gas is in one container, that is ordered. If it's in both containers, that's more random. Us as living organisms, we hate randomness, because to have our cells, to have our DNA, to have our proteins the way it's made, that is incredibly ordered. Living material is incredibly ordered. And we need to eat food to make sure it's ordered. Once you stop eating food, your cell goes back to disordered and you die. So when you die, your body goes back to that disordered randomness. Again, two different terms. We really talk about this. Entropy, think of this as chaos, randomness. Don't worry about the equation there. I'm not going to ask you about Boltzmann constants. I like them, but we don't need to know about that. Enthalpy, heat. We really look at this in types of bonds. And we combine these two together to something we call free energy, or Gibbs free energy. So that G stands for Gibbs. And the change in Gibbs free energy is equal to the change of enthalpy, or heat, or bonds being broken, formed. So that's for bonds, minus temperature, Kelvin, change in randomness, or entropy. And delta G is really going to tell us if a process is spontaneous or not. So delta G has a lot of different definitions. One, its ability to do work. Two, it tells us if it's spontaneous. And so delta G can have three values. It can have negative, 0, or positive. Negative is spontaneous. 0 is equilibrium. Positive is non-spontaneous. So the last thing I'm going to just do here is give you a definition of spontaneous versus non-spontaneous, as that concept's actually not like, it's not easy to think about. So my system is going to be this pen. And I'm going to drop this pen. And because of the laws of physics and we're on Earth, that pen's going to drop. That's spontaneous. Sorry if you have your headphones on, that was loud. So the pen moved to the ground by itself. I didn't put in any energy to make that happen. That's spontaneous. Okay, now I'm going to have my pen jump back into my hand and go. And go. So the pen jumping back to my hand is not spontaneous. If I want it to come back to my hand, I go and pick it up. I use my energy to move my muscles and pick up my pen. So if something is spontaneous, it happens without more energy being invested. If it's not spontaneous, you have to put energy in. If a spontaneous process goes one direction, the opposite of that process is always non-spontaneous. Dropping my pen, spontaneous. Flying back to my hand, non-spontaneous. Right now, although you can't see it, the pen is on my desk. It's not moving. It's at equilibrium. It's happy. As long as no outside force touches it, that pen will remain on my desk until my desk decomposes in a couple thousand years. So we're at equilibrium right now until I move it. So yeah, that's what spontaneous and non-spontaneous means. And on Wednesday, we're going to get into the calculations of thermodynamics. And with that, that is the end of our first class. We will continue on Wednesday. So just as a reminder, I will put up a homework that will just ask the same type of questions we went over in class today. Those are due next week, Monday at 8 a.m. For Wednesday at 8 a.m., you have that reading guide due. So please watch the videos. Please fill out the reading guide. Turn that in. And as long as you answered all the questions and you put in effort, you get 100% on that. And if you do those every semester, that's a free 6% towards your grade. But yeah, that's all I have for you. Thanks for coming by. If you have any questions at all, email me. Otherwise, I'll see you on Wednesday. Take care, everybody.